All right, class, today we're going to start lecture 2 of unit 4, and this is where we're going to talk about protein digestion throughout various animals. So first we're going to start with protein digestion in non-ruminants because, as you'll see later, with ruminants we're mainly having to deal with microbial digestion. So the actual protein digestion by non-ruminants or ruminants, once it gets into the GI tract, is pretty similar. So our protein digestion and absorption in non-ruminants, basically the proteins are digested by hydrolysis. We denature the protein with both chemical through the hydrochloric acid in the stomach, and we have enzymatic digestion that starts in the stomach. So all protein digestion starts in the stomach, contrast to carbohydrate, specifically starch, and starts in the mouth with most species. The digestion continues through the intestinal lumen and also at the enterocyte membrane, just like with the carbohydrate disaccharidases that are found on the enterocyte membrane. We also have peptidases that are found on the enterocyte membrane. Our proteins are absorbed as very small peptides, so we have di and tripeptides, and they're also absorbed as single amino acids by the enterocytes, and they're transported through the enterocyte. So although the mouth doesn't have specific enzymes like salivary amylase for starches, it still has some function. So the mouth is actually moistening and crushing protein structure in the food or feed. But the actual digestion begins in the stomach. So it begins in the stomach. And this first starts with the gastric juice. So if you remember, we have our 2 to 5% hydrochloric acid, which drops our pH down to about 1 to 2. And this hydrochloric acid is denaturing the protein, so we are breaking the hydrogen bonds. It also hydrolyzes portions of the protein. So that's what our chemical component of our protein digestion is doing. And then we have proteases, and one that we've learned about in the first unit is pepsin. So pepsin is released as pepsinogen, and it is an endopeptidase, meaning that it is cleaving the inside of the peptide, and it's specific for aromatic and dicarboxylic amino acid residues. So it is very specific into which ones it's going to be breaking in cleaving. So things that I want you to take home from this slide related to the proteases is pepsin is an endopeptidase, and it's specific for aromatic and dicarboxylic amino acids. So you may see a true-false saying that pepsin is a exopeptidase specific for aromatic and dicarboxylic amino acids, and that would be false because it's an endopeptidase. So although protein digestion actually begins in the stomach, that actually results in very little overall protein digestion. We need that to initially get everything started to break that tertiary structure and quaternary structure in some of the secondary structure to where we get these very long peptides. But the vast majority of our protein digestion is actually due to what is released from the pancreas. So our pancreas is secreting proteases into the duodenal lumen, And these proteases are breaking the protein up into tri, which is 3 amino acids, di, which is 2 amino acids, and then we have single amino acids. Our enterocytes, as I mentioned before, they also have proteases located on the surface of the enterocyte. So there are proteases located on the surface. And these are basically going to be breaking up some of these di and tripeptides and some of the shorter but still oligopeptides, which is more than 3. And we're going to be getting single amino acids and dipeptides are going to be the main things that the proteases on the surface are going to be breaking up. And then our enterocytes, they are absorbing single amino acids, tripeptides, and dipeptides. And up until about 15 years ago, it was thought that all amino acids were absorbed as single amino acids, because if you looked at the amount of amino acids found within the food, and then you look at what is in the blood right after the intestine. So as soon as it enters the blood, we had a bunch of single amino acids entering the blood. Well, it actually turns out that inside the enterocytes, they have proteases that are breaking up those di and tripeptides. So these enzymes that are released from the pancreas, these proteases, are either endopeptidases or exopeptidases. So if we have our protein that is coming in from the stomach, so this is actually more of peptides here. And as those peptides enter the duodenum, we're going to have our CCK stimulated to stimulate the pancreas to secrete the pancreatic juice, which includes trypsinogen, chymotrypsinogen, and proelastase. So these are all endopeptidases in that they are cleaving in the middle. So they are enzymes that are breaking peptide bonds from inside the peptide. And each one of these proteases are specific. So our trypsin is looking for the basic amino acids, and it is cleaving on the carboxyl terminal end of those amino acids. Our chymotrypsin is cleaving the aromatic amino acids, and it's also going to be cleaving on the carboxy terminal end. Our elastase is looking for the aliphatic amino acids, and it is going to cleave also on the carboxy terminal end. So these are all endopeptidases. So it's looking for these basic aromatic or aliphatic amino acids, and that's where they're going to cleave on the carboxy end of those. In contrast, we have exopeptidases. So these are breaking peptides on the outside of so they're going from the terminal ends. So our carboxy peptidases are cleaving on the carboxyl end. Which is our COOH end. And we have two different types. So we have carboxypeptidase B and carboxypeptidase A. So our carboxypeptidase B is going to be taking and cleaving where we're going to get arginine and lysine. So if arginine and lysine is on the terminal end of the peptide, it's going to be breaking those off. So our products are arginine, lysine, and then we get small peptides. If there's next one is an arginine or lysine, carboxypeptidase B has to leave it alone. And next we have our carboxypeptidase A. And this is looking for neutral amino acids. So any of our neutral amino acids, if they are on the end, carboxypeptidase A is going to be cleaving those. So we're going to have those as our single products, and then we also get small peptides. There are also aminopeptidases, so they are going to be working. So they are also exopeptidases, and they're going to be working from the amino end. So this is basically reiterating what we just talked about, just in bullet point form. So summarizing, we have our endopeptidases, which we have our trypsin, our chymotrypsin, and our elastase. So I want you to be able to tell me that these three are endopeptidases, and also include pepsin. Pepsin is an endopeptidase, but that's coming from the stomach. So trypsin is cleaving on the carboxy side of our basic amino acids. So remembering to the last lecture, that's going to include lysine and arginine. Our chymotrypsin is cleaving on the carboxy side of our aromatic amino acids, including phenylalanine, tyrosine, and tryptophan. And our elastase cleaves peptide bonds between glycine, alanine, valine, and phenylalanine. With our exopeptidases, they are carboxypeptidase. They cleave all the amino acids on the remaining fragment of the one amino acids. And like I said, we have two different types. We have carboxypeptidase A and carboxypeptidase B. We also have aminopeptidases, which these are cleaving amino acids from the amino terminal end. So we have to regulate our protein digestion. So one of the main ones that we need to regulate is trypsin. So the trypsin is basically going to be activated, and it's going to activate the other zymogens that are released from the pancreas. So we have hormonal regulation of trypsin secretion. So if you remember back from the previous units, we had mentioned about secretin and cholecystokinin. So basically we have the pancreas. When we have protein entering the duodenum, we have our endothelial cells that have receptors for the protein, and they're going to sense that protein entering the duodenum, and that's going to signal those cells to release CCK and secretin. So once protein enters the duodenum, we're having CCK and secretin secreted, and that's signaling to the pancreas to release the gastric juice. Now, we also have intracellular inhibition of trypsin, and then this is basically due to the fact that we want to neutralize any activated trypsinogen. So we release trypsin in the form of its zymogen, trypsinogen, but nothing is 100% foolproof, and there are errors that occur. We always like to make sure that we are able to control for anything that happens. So we don't want to start digesting ourselves. So we have intracellular trypsin inhibition that the cells have to neutralize any activated trypsin that we did not want activated. So how is trypsin originally activated? So we have an enzyme called enterokinase. So be able to tell me this, that we have enterokinase that activates trypsinogen to trypsin, and it cleaves a hexapeptide from the amino terminal end. And this enterokinase is actually thought to be species specific, whereby you can't take, for instance, swine enterokinase and have it activate chicken trypsin. So it's somewhat specific for each species. And then once we have activated trypsin, it goes on and activates itself So then we end up having enterokinase and trypsin activating more trypsin, and trypsin is also what is needed to activate chymotrypsin. So we have trypsin cleaves off and activates trypsinogen, so it forms chymotrypsin. So that's how we activate trypsin. Now we also need some ways to control the secretion of trypsin. As I mentioned, we don't want a bunch of trypsin entering from the pancreas, and then eventually we get auto-digestion of our own cells and protein. So we have what we call monitor peptide. Our monitor peptide is actually released with the pancreatic juice. So it's released, stimulated by cholecystokinin secretion. And we'll talk about that a little more in the next few slides. And then we also have some exogenous inhibition of trypsin. So the most notably known is trypsin inhibitor found within soybeans. And this actually ends up inhibiting the action of trypsin, and that decreases the protein availability. So this is a way that soybeans have evolved to, because they have such high protein content, that they wanted to protect themselves and decided that this was the best way to limit and keep animals from consuming too much of it. However, we are able to denature this in a feed mill setting to where we cook it, and then the soybean ends up getting denatured. So more specifically, what is monitor peptide? So this is a peptide found within pancreatic juice. So it's secreted along with the trypsinogen, chymotrypsinogen, etc. And it actually binds trypsin. And it causes the release of more cholecystokinin. So it interacts directly with the cholecystokinin producing cells, and the cholecystokinin ends up stimulating the release of more trypsinogen. So basically, if we have free monitor peptide, it ends up signaling that we still have a lot of protein available that we need to digest. So we end up secreting more of the trypsinogen to help break down all that protein. And if we end up inhibiting this monitor peptide bonding, we're going to keep on having, so if we make it, for instance, with the soybean trypsin inhibitor, so our soybean trypsin inhibitor is binding protein, so that's giving a false sense of having free monitor peptide, and that causes our free monitor peptide to signal the cholecystokinin production. So we constantly keep on having more CCK signaled, and therefore we have more trypsinogen secreted into the lumen. So here we have a schematic of what's going on. So we have our protein and amino acids are coming into the lumen. So this is inside of the lumen of the duodenum. These are going to signal cholecystokinin release. So CCK is released. We have an effect on the pancreas, and we're going to be secreting trypsinogen, which then gets activated to trypsin. When we have our trypsin, our monitor peptide is competing with the protein and the amino acids for the trypsin. So if we do not have any protein in the diet, let's say you ate pure Kool-Aid, sugar that you can get from the grocery store already with the sugar in it. So we don't need any trypsin when we're consuming that. So basically we're going to have our monitor peptide is going to be able to bind the trypsin. So our active monitor peptide that comes from the pancreas binds the trypsin, and that becomes inactive. And when we have it inactive, that is inhibiting here our CCK release. So we have no CCK that's released when we have monitor peptide bound to trypsin. Monitor peptide plus trypsin is inactive. However, when we have adequate amounts of protein, we're going to end up having our trypsin is going to bind greater with our protein and amino acids. So that's going to have a bunch of free monitor peptides. So if we have free monitor peptide, that's meaning that we don't have enough trypsin. We need to start breaking down more of this protein. We need more trypsin. So that's where we're going to have a positive effect on our cholecystokinin. So our protein is competing with our monitor protein, and any unbound monitor peptide stimulates the cholecystokinin release, and that stimulates more secretion of trypsinogen. So you can see here, if we have trypsin inhibitor within our food or feed, so if you eat a lot of soy, let's say you really love raw soybeans, that aren't cooked. So we're going to have our trypsin is going to bind to our trypsin inhibitor from our soybeans. So that's going to simulate the trypsin being bound to the protein. So that's going to end up having more of this monitor peptide in its active form, stimulating more cholecystokinin release. And then we're going to end up getting more trypsin because your body is sensing that, well, we still have a lot of protein here that we need to break up. But that actually ends up causing hypertrophy of the pancreas, which we'll show in a few minutes. So as I mentioned before, we have these trypsin inhibitors, and trypsin is the most critical one out of all the proteases released from the pancreas. It's the one that's activating everything else, so it's the main regulator. If we can control trypsin, we can control all the other proteases. So these inhibitors are small molecular weight proteins or peptides that inhibit trypsin. So as I mentioned before, we have endogenous ones. These are produced by the cells to protect any activation of trypsinogen that if it was activated accidentally. Then there are also exogenous ones, which is the soybean trypsin inhibitor. So if we have any and those plants have evolved to make those because they have high protein content in their seeds. So they wanted to make sure that they were going to limit the amount of consumption that animals do to the plants. So let's discuss the soybean trypsin inhibitor a little more. So raw soybeans have a very high level of trypsin inhibitor. And if you feed raw soybeans to animals, you see a depressed growth in rats, chicks, mice, pigs. And that's basically because you're depressing the protein digestibility, and you get pancreatic dysfunction because you have all that free active monitor peptide signaling CCK, and you release more and more trypsin from the pancreas. And because of that, you cause hypertrophy of the pancreas. Thankfully, trypsin inhibitor is not very stable, so it can be degraded or denatured very easily with cooking. So a lot of times the feed mills and various food processors, they will check the amount of trypsin inhibitor, and they actually utilize a rapid test called a urease assay. So this urease enzyme is highly correlated with the trypsin inhibitor. So this is a very quick assay that you can do, and if there's a lot of trypsin inhibitor, then the soybeans become a very bright, almost fuchsia, purpley pink color. And if you have complete denature of the trypsin inhibitor, it will just look like wet soybeans. They're typically a light brown, yellow color. So we actually want to retain some of the trypsin inhibitor found within soybeans to where it's between 5 to 20%. And this is because if we get rid of all the trypsin inhibitor, we end up denaturing all the protein, and then that makes it to where it's less available for the animal to utilize. So we don't want to overcook our protein source. So we need to have it just right. Our raw soy flour contains 52.1 trypsin inhibitor activity units per gram of sample, where if you cook it, we're able to bring that down to 3.2 to 7.9. And trypsin activity is decreased within the intestines when trypsin inhibitor is present. And basically, the less monitor peptide is inactivated, indicating that there's a trypsin shortage, which is what's causing the hypertrophy of the pancreas. And you get more CCK produced because of the presence of the unbound trypsin inhibitor, and you get an overproduction of trypsin. So you can get pancreatic hypertrophy or pancreatitis. So here we have our soybean trypsin inhibitor. Our trypsin inhibitor is green here in both pictures. So here we have our trypsin. So our white is our trypsin. Our green is our trypsin inhibitor. And basically the way it works is we have this lysine moiety in red here, and this lysine component is able to bind into the active site of trypsin and basically blocks all of the action of trypsin, and it ends up staying bound. So basically you end up having the trypsin, and the trypsin inhibitor ends up being excreted in the feces along with all the undigested protein that is available. So this trypsin inhibitor really limits the digestibility of the protein within soybeans. Okay, so now we're going to switch gears a little bit and talk about how peptides and amino acids are absorbed. So as I mentioned before, you do have some peptides that are able to be absorbed from the enterocyte, and this is relatively new within the last decade or two that this has been discovered. But it was always thought that amino acids had to be single amino acids when they were absorbed, and that was just based off of looking what we had within the lumen versus what was entering the blood. And in the last decade or two, we've actually discovered that there are intracellular peptidases, and then actually 60% of the protein is absorbed as peptides, so di and tripeptides mainly. And peptides that have high amounts of glycine, proline, hydroxyproline, and dicarboxylic amino acids those ones are at a greater rate hydrolyzed in the cell. So here we have our schematic of our peptide and amino acid transport. So here we have our protein on the right. It comes in, we break it down by pepsin and trypsin, and we end up getting our free amino acids. We have large peptides and we have small peptides. So our trypsin, chymotrypsin, elastase, our carboxypeptidases, and aminopeptidases are going to be breaking down our protein to large peptides and some free amino acids and small peptides. We do have these peptidases that are located on the microvilli of the enterocyte. So these peptidases are resulting in, we get free amino acids and we get small peptides. So our free amino acids, they can come in through their transporters. So we have neutral, basic, and acidic transporters, and they are able to enter in. And there are many different transporters found on the microvilli, and so we're not going to get into all the different types. And then we have small peptides. So these small peptides are actually able to come through a carrier called pepti1. And once these small peptides enter through the pepti1, we have these, they are broken down by these intracellular peptidases, and that's where we're getting our single amino acids. And then those are single amino acids that came in directly through the transporters from the lumen, and then our amino acids that were broken down by our intracellular peptidases. Both of these are able to enter the blood. So you could see where it baffled scientists and where we thought for the longest time that all of them had to come in through is free amino acids. But that is not the case. This is actually the primary way, so 60% of our amino acids are coming through our pepti1, and that leaves only 40% are coming through our free amino acids. So our single amino acid transporters, if you remember back, I believe in the second unit, maybe even the first unit, where we talked about how they are, the various macronutrients are absorbed. So amino acids are utilize active transport, meaning that it requires energy. So similar to glucose, it utilizes a sodium-dependent chemical gradient, so it utilizes the sodium-potassium pump also. And our peptide transport, this is going through pepti1, and it is proton-driven, so we exchange it for hydrogens. And we can transport between 400 different dipeptides and 8,000 different tripeptides. And doing the math, we have 40% of protein is absorbed as single amino acids, resulting in 60% coming through as the peptides. So as I mentioned before, we have the different transporters, and they are transporting based off of the R group classification of the amino acids. So we have our acidic amino acid transporter, so this includes glutamic acid and aspartic acid, so those two are sharing the acidic amino acid transporter. Our basic amino acid transporter, which transports lysine, arginine, and histidine. Our neutral is looking for any of her neutral amino acids, including alanine, glycine, and serine. And then we have another component which transports glycine, proline, and hydroxyproline. And so since these amino acids share a lot of these transporters, we end up getting what we call amino acid antagonism. Where these transporters will actually have a greater affinity for a particular amino acid over another one. So for instance, if we were to have a diet that is adequate in arginine, but we overfeed lysine, we are going to cause an arginine deficiency because this basic amino acid transporter prefers lysine over arginine. So it has a greater affinity for lysine. So inadvertently, even though we had adequate amount of arginine within the diet, we overfed lysine, so we are going to cause an arginine deficiency. So that's what those amino acid antagonisms are. So not only is it important to, as a nutritionist, to understand that yes, we've had these amino acids are meet or exceed the requirement, but we also want to make sure we have all of them in the proper ratios, knowing that they are sharing transporters, and consequently we're trying to avoid any sort of induced deficiency. So our main peptide transporter is PepT1. So peptides are absorbed typically as di and tripeptides. You can get a few tetrapeptides, which is 4 amino acids long. But PepT1 is the main transporter for these peptides. It is membrane bound. It has a sequence of about 708 amino acids. You don't have to know that at all. But here you could see when I said it's proton driven, so we have our amino acids come in, and we also, you can see we have this hydrogen that needs to come in with it. And then we have to pump that hydrogen back out. And that hydrogen is pumped out, and we bring the sodiums in, and that's where we end up using our sodium-potassium pump to alleviate and keep the gradient. You do also have some peptides that are able to, they're endocytosed into the cell, and so within the cell they end up, that's how they get in, and then you'll have the intracellular proteases that are breaking those down. And sometimes there's a small amount of the peptides that can be found within the blood, and it's thought that those are coming from the tight junctions. So in between the two cells, they're able to enter in. So up until now, we have focused on protein digestion and non-ruminants. So now we're going to look at how protein digestion differs or is similar with ruminants and horses. So protein digestion with ruminants is pretty similar. The biggest thing is, so we have to deal with the rumen. So the microbes get the first shot at all the protein that comes in. However, once the digesta enters the abomasum, the fourth compartment of the ruminant stomach, which is equivalent to our true stomach, we have the secretion of hydrochloric acid, pepsinogen, and then they also have renin. So renin is an enzyme that's found within the gastric secretions of calves and lambs, and it converts milk protein and calcium to calcium pericaseinate. So this is, if you're ever eat cottage cheese, this is the curd. So basically this is the renin actually makes the casein insoluble. So that's why you get this formation of the curds. And we have commercial renin, which is called rennet, and that's actually prepared from the abomasum of calves. And then we also have recombinant microorganisms that we utilize to make the commercial rennet. So this is very important for people that make cheese. They utilize a lot of rennet. And interestingly, if you go back thousands of years, they used to utilize the abomasum as a carrier for milk for individuals that were going on long trips. And that's how they actually first discovered the rennet, because after a few hours, that milk within the abomasum was able to form a big curd within the abomasum. So then it provided the traveler some protein, and they were able to drink the whey component and eat the casein. So this is a schematic of the casein micelle that was proposed by Wall in 1970. So you could see that the casein is made up of different chains we have an alpha chain, kappa chain, and a beta chain. And our rennet is used, it breaks the kappa chains and the kappa casein and the alpha casein. And when we break those chains, we're able to allow for an influx of calcium to enter in. So we have a bunch of calcium that's able to go into the inside of this casein micelle. And that's what's allowing it to precipitate. So if you remember the old nursery rhyme, Little Miss Muffet sat on a tuffet eating her curds in whey. She was eating cottage cheese, and that was all made from rennet. So you have milk exposed to rennet, and it's going to break the kappa casein and the alpha casein. We have calcium that's able to infiltrate, and that causes it to precipitate out. So the whole reason why the calves and lambs do this is to slow the transit time of the milk for the animals. So they want to make sure they have adequate time to digest all of this, and so they evolve to slow down the transit of the milk, as it is one of the most nutritionally complete food sources out there. And if you remember, if we keep the abomasum full, that's going to reduce the amount of ghrelin that's secreted, causing you to be hungry again. So this is actually what is how that slows the transit time. And then we have the rennet, which is artificially synthesized from microorganisms, and that's used by cheesemakers. You still have some old-timey, old-fashioned cheesemakers that actually prefer to use the rennet that is isolated from the abomasum of calves and lambs. They believe it provides better flavor, etc., to their cheese. If it does or not, I'm not really sure. But most of, especially American cheese that we're eating here, unless you're getting really fancy, we're all utilizing the genetically modified bacteria to make the rennet. So how does microbial digestion affect ruminants and hindgut fermenters? So for our ruminants, the microorganisms degrade a large percentage of the dietary protein, and this depends on the characteristics of that protein. Now, some ruminant nutritionists are trying to find what we refer to as bypass protein sources. So as I mentioned before, that ruminants don't have an amino acid requirement because we're not able to control which amino acids they are or are not able to absorb, because we don't necessarily know the proteins that the microorganisms are switching the amino acids to. So some researchers have been looking into developing what we refer to as bypass proteins, where it can bypass the rumen and then be degraded just like a non-ruminant. But the most part, the ruminal protein is a great percentage of the ruminal protein is degraded by the microorganisms, and they break it down into ammonia and carbon. Ammonia is the favorite nitrogen source of rumen anaerobes, and they utilize the carbon and the nitrogen or ammonia to form new microbial amino acids and protein. Now, you can provide ruminants other non-protein nitrogen sources, such as urea. You can feed them urea, you can feed them poultry litter, anything that has high concentrations of nitrogen. Now, you have to be careful and make sure that you don't overfeed the non-protein nitrogen, because you may, the limiting effect is how quickly the microbes are able to convert it to protein, so we don't want to overrun them with a bunch of nitrogen. And then our post-ruminal digestion, very similar to that of non-ruminants. So here we have a schematic of what protein digestion looks like in the ruminant. So here we have our true protein and our non-protein nitrogen that both enter the rumen. Of this, we have 40%. So be able to tell me that 40% of the protein is able to escape the rumen and go straight into the abomasum and then be digested like is accustomed in non-ruminants. However, the other 60% is broken down to ammonia, and then the ammonia ends up being converted to is absorbed through the rumen, brought to the liver, and is then secreted back in the saliva. And so you have this recirculation of nitrogen due to this being put back into the saliva. So we're always providing microbes with adequate amounts of nitrogen so they can make the microbial protein for us. So as I mentioned before, our non-protein nitrogen, or NPN, can be fed to ruminants. So this is usually, we utilize urea as the main form of non-protein nitrogen that we feed. And this we do because we're able to spare the dietary protein. So protein is expensive, urea is cheap, and so we just put in a little bit of urea so we reduce the amount of dietary protein. The problem is if we end up feeding too much non-protein nitrogen, we end up getting ammonia toxicity. And we have two levels of ammonia toxicity. So one is subclinical, where you get subpar growth and performance. So the animals just aren't growing as well. They may not be producing as much milk or laying, these are dairy. So milk, they would be producing less milk. Our replacement heifers would not be growing as well. Our steers, etc., will not grow as well. So subclinical, anything that's less than 300 micrograms per milliliter of blood. Now, if you have really bad TMR that is provided, that maybe it was off by a decimal point, so now you've put in a lot of urea. So you're going to get clinical ammonia toxicity. So this results in you get staggering, frothing at the mouth, convulsions, and you'll also see them doing head pressing. So if the blood ammonia levels are greater than 1,000 micrograms per milliliter, that is very clinical ammonia toxicity. And this is all because ammonia is affecting the central nervous system. So ruminants, for the most part, or any of our foregut fermenters, are more susceptible to our ammonia toxicity than our non-ruminant hindgut fermenters. However, you could see here, due to this picture of the horse doing some head pressing, that they are still susceptible to it. So why do we get these clinical signs? Why is there head pressing and convulsions, etc.? Basically, so the ammonia is able to be metabolized in the brain, so it's able to cross the blood-brain barrier, and we have the alpha-ketoglutarate is converted to glutamic acid, and it forms glutamine when ammonia is present. So basically, we have our TCA cycle. Our alpha-ketoglutarate is being pulled out of the TCA cycle to help form glutamine so we can clear the ammonia. So basically, we impair our ATP production in the brain, so that's where you're getting convulsions. Basically, the brain is saying that I don't have enough energy, and the convulsions are being signaled because we need more glucose secreted so we can get more ATP through the TCA cycle. But that's not going to be helping because we keep on pulling more of the alpha-ketoglutarate out to deal with the ammonia. So now we're going to spend a little bit of time talking about protein digestion and the hindgut fermenters. So how is nitrogen metabolized in the hindgut? Well, it's pretty similar to what we would encounter in our foregut fermenters. So any nitrogenous substance that enters the hindgut is subjected to microbial digestion. Typically, we end up this microbial digestion of the protein, and we can get some toxic amines that are produced. We also have some components that have pharmacological effects. So we get vasoconstriction, we get cell proliferation. So the byproducts, depending on the protein that's broken down or the amino acids that are broken down, the byproducts that the microbes are making are going to cause vasoconstriction or cell proliferation. So this is one of the things with laminitis. So if you've had horses that get into a bunch of grain and get overconsumed, and they've had it for a long time, they'll get laminitis. Not only is that due to the volatile fatty acids that are produced, and then you have the issues due to that, but we also have the protein that enters the hindgut is going to end up causing vasoconstriction. So that's where you're going to reduce the amount of blood going to the hoof. We're also going to get cell proliferation that is activated. So then we're going to have an over growth of the hoof, etc. And then the digestion of the microbes is very much produces odoriferous compounds. So each animal has its own characteristic odor. I'm sure you've ever been to various farms. You can distinguish very quickly a pig farm from a chicken farm, from a horse farm. So all of that has to do with the compounds that the microbes are producing in the hindgut. And typically, our carnivorous animals are more odoriferous than our herbivores. So if you think comparing a dog is a little more stinky than, let's say, a rabbit. Now, as an animal nutritionist, we want to make sure that we want to try and limit the amount of protein that is entering the hindgut, because we want that high, good quality protein to be utilized by the animal, not necessarily by the microbes, especially in our hindgut fermenters. Unless they are practicing coprophagy, we're losing out of a lot of the benefits of that hindgut fermentation and microbial protein that's developed. So a lot of specifically the poultry industry and the swine industry, they utilize what we refer to as standard ileal digestibility. So this is where we balance diets based off of the amount of protein that's entering the hind gut. So we want to really reduce the amount of protein entering the hind gut for a few reasons. One, it's going to help reduce the fecal odor. As I mentioned, protein's very expensive. It's the second most expensive component of animal diets. So we want to make sure that the animal is utilizing that, not the bacteria. In addition, it's going to help reduce nitrogen pollution. So currently there's a lot of regulations on livestock production looking at nitrogen runoff into lakes, rivers, and streams, etc. So if we formulate diets based off of what the animal is actually absorbing, and we can reduce the amount of excess that's going into the hind gut and being utilized. So there are a few ways in which we can do this, but most commonly we put in a cannula right at the ileocecal junction. So we surgically cannulate the animal, and here on the lower right you could see a couple different schematics of the cannulas. So here we have what we refer to as a T cannula. So this comes in here and it's basically a metal component, and it's a T, so you have a stopper at the end, and the digesta moves through. You pull out the stopper and you can have collected in a bag. We also have a PVTC one that you can see it here. And then we have, and these two are less common, then we have the re-entrant cannula, to where we basically have two T cannulas, and we can direct the digest and we can pull out from here. But for the most part, the T cannulas are the most common, and you can see this done in horses, pigs, and cattle. In poultry, we do not typically put in a surgical cannula. If you remember that the colon in the chicken is relatively small, so it's actually, we just remove the ceca. So we surgically go in and remove the ceca, and the digestion does not spend enough time within the large intestine, so we just take whatever is in there. The microbes aren't going to have a significant contribution to any of the digesta or the standard ileal digestibility. So that's what this is, and this is really what we're moving towards. As I mentioned, swine and poultry industry uses the standard ileal digestibility to formulate diets, and other livestock species are getting there also.